Welcome back to this next video, and in this video we are going to talk about the G alpha Q signaling pathway, which actually works through the IP3 and DAG pathway. Now this is a series of videos in which we are focusing on the serine threonine kinases, and this G alpha Q subunit of the heterotrimeric G protein is actually responsible for the activation of this protein kinase C, which is a serine threonine kinase utilizing the IP3 DAG pathway. Now this is the whole story that we'll be focusing on in the series of videos that we are doing on the protein kinase C. Now what happens is there is a G protein coupled receptor, for short, which is known as the GPCR. This G protein coupled receptor, on its interior side, on the cytosol side, a G protein is attached to it, and this G protein is actually a heterotrimeric G protein. What I mean by that is that the heterotrimeric G protein is made up of three different subunits. One is known as the alpha, the other is known as the beta, and then you have got this gamma protein. Now, in this particular video, we are going to talk about a special kind of the alpha subunit, which is known as the GQ subunit. So when a ligand, say for example the acetylcholine, it binds to this G protein coupled receptor, initially to the alpha subunit, a GDP molecule is attached. But after the binding of the ligand, there are conformational changes taking place in the G protein coupled receptor, and this G protein coupled receptor is responsible for replacing the GDP of the alpha subunit into a GTP. So what you are getting is you are getting an alpha GTP. When the alpha subunit is attached by a GTP, it actually leaves the beta and the gamma subunit, and this alpha GTP go and binds to a particular enzyme which is known as the Phospholipase C. Now what this Phospholipase C do is that it go and degrade a particular phospholipid in the cell membrane, which is known as the PIP2. So when the protein lipase C, it gets activated by the alpha GTP, it go and it break down the PIP2 into the DAG, which is actually the Acyl Glycerol, and into a soluble molecule which is called as the IP3. When this IP3 that is released, it go and it binds to a special receptor on the endoplasmic reticulum, and after the binding of the IP3 to this special receptor, the calcium that is actually moving from the endoplasmic reticulum into the cytosol or into the cytoplasm. Now some of this calcium, it is responsible for the activation of the protein kinase C, and then this protein kinase C is actually going to perform its various activities. So this will be the whole story that we'll be focusing on in the coming videos and in this particular video. So the first thing is, what is a G protein coupled receptor? Now, if you talk about the G protein coupled receptor, they are also known as the 7-transmembrane protein. What this means is that if you look at the structure of the G protein coupled receptor, it is a single protein. This one is the amino terminal, this one is the carboxy terminal, and this particular G protein coupled receptor is spanning the membrane 7 times. This is the spanning number 1, which is known as the transmembrane domain 1. Then it span the membrane second time, which is known as the TM2, and so on until it passes the membrane 7 times. So this is known as the 7-transmembrane protein as well. Now, when the G protein coupled receptor, it spans the membrane 7 times, it is creating special loops on the inside and the outside of the cell. On the outside of the cell, that means on the extracellular side, it is actually having these extracellular loops. In 3 of them they are there. One is known as the extracellular loop 1, the 2, and then the 3. And this extracellular loop is actually responsible for the binding of the ligand. The loops that are made on the cytoplasm side, they are known as the intracellular loop, and they are for short known as the ICL. and just like the extracellular loops, there are 3 intracellular loops. One is known as the ICL1, the other one is the ICL2, and the ICL3. And these intracellular loops are actually providing a binding site for the G protein. Therefore, they are known as the G protein coupled receptor. So if you look at in this particular diagram, on the interior side, on the intracellular loops, the G protein that binds in its heterotrimeric form. Now, initially, when there is no ligand, these heterotrimeric G protein, they are actually in the trimeric form. That means that the alpha, beta, and the gamma subunit, they are attached to each other, and a GDP molecule, it is attached to the alpha subunit. When the ligand, it binds to the G protein coupled receptor, the G protein coupled receptor goes through the conformational changes, and the GDP of the alpha subunit, it gets replaced by a GTP, and when it's replaced by the GTP, it actually gets dissociated from the beta and the gamma subunit, and this GTP form of the alpha subunit is then responsible for creating a signaling pathway. So this is the first part of the pathway, which is the G protein coupled receptor. The second important thing that I want to focus on is the type of the alpha subunit, because in this particular video we are actually focusing on a single type of the alpha subunit. When you talk about the heterotrimeric G protein, there are broadly 4 types of the alpha subunit. Now, one type, they are known as the G alpha I, and these are responsible for the activation of a specific set of responses. Then you have got this G alpha S, and I have a detailed video on the G alpha S. I will share the link in the description. So this is the second type of the alpha subunit. And the third type of the alpha subunit is known as the G alpha 12 and 13. And the one that we are focusing on in this particular video, which is responsible for the activation of the protein kinase C, that is known as the G alpha subunit. So all of these heterotrimeric G protein and their alpha subunit works by the same mechanism, that after binding of the ligand, their GDP, it gets replaced by the GTP, but of course they are producing a specific set of response. So we are actually focusing on this particular subunit, which is the G alpha Q alpha subunit. Now, when the ligand, say for example the acetylcholine, it binds to the G protein coupled receptor, and the alpha subunit, it gets activated, it is then going for the activation of the enzyme, which is known as the Phospholipase C. In this particular case, the specific type of the Phospholipase C that we are talking about is known as the Phospholipase C beta. Now, what this Phospholipase C do is that when the alpha subunit containing the GTP, it binds to the Phospholipase C, it gets activated, and the Phospholipase C then go and acts on the Phosphatidyl inositol 4,5 bisphosphate. Now, what this Phosphatidyl inositol 4,5 bisphosphate, or for short, which is known as the PIP2, how it looks like. Now, this is the cell membrane of the of a cell. For example, this is the cell membrane, and the inside the cell membrane, this particular structure that I am showing you, you should consider that between this cell membrane, it is the interior of the cell membrane. So inside the cell membrane, there are long chain fatty acids which are attached to the glycerol molecule. So this is a glycerol having 3 carbons over here, and to the 2 carbons of the glycerol, long chain fatty acid, they are attached. Now, this particular structure, the glycerol and the 2 long chain fatty acid attached to its 2 carbon, it is known as the diacyl glycerol. So the long chain fatty acid plus the glycerol, it is known as the diacyl glycerol, because there are 2 acyl or 2 long chain fatty acid attached to it. To the 3rd carbon of the glycerol, a phosphate group that is attached. So if you combine this whole structure, the long chain fatty acid, the glycerol plus this phosphate, it is known as the phosphatidyl. So this particular term, this phosphatidyl, it actually refers to the 2 long chain fatty acid, the glycerol, and a phosphate group attached to one of the carbon of the glycerol. Now, the next thing in the name of this phosphatidyl inositol is the inositol. So this inositol is actually this 6-ring carbon structure. This particular 6-ring carbon structure, as you can see, this is 1, 2, 3, 4, 5 and 6. This particular structure is known as the inositol. If you talk about this 4,5 bisphosphate, so if you look at the inositol at position number 4, there is a phosphate group. At position number 5, there is a phosphate group. So it actually makes the inositol 4,5 bisphosphate. So this bis actually refers to 2 phosphate groups. So this is the complete structure of the phosphatidyl inositol 4,5 bisphosphate. Now this particular structure, this whole structure, the long chain fatty acids and the glycerol, that is actually present in the cell membrane, that is not outside the cell membrane. This particular structure, you should consider that within this cell membrane. And then this particular structure, the inositol bisphosphate structure, is actually present in the cytoplasm of the cell. Now when the phospholipase C beta, it gets activated by the alpha GTP, what it do is that it is going to make a cut at this particular position where the glycerol, the carbon of the glycerol, that is actually interacting with the phosphate over here. So the activated phospholipase C beta that cleaves over here utilizing a hydrolysis reaction. And when you go for the hydrolysis, actually a hydrogen group that is attached at this particular position. So when you cleave the PIP2 or the phosphatidyl inositol 4,5 bisphosphate at this particular position, you are going to get 2 things. One is known as the diacyl glycerol, that as I've told you, and this diacyl glycerol that is going to stay in the cell membrane, that will be still attached to the cell membrane. Now this particular molecule that is produced is known as the inositol 1,4,5 trisphosphate. So if you look at the difference between the inositol 4,5 bisphosphate and the inositol 1,4,5 trisphosphate is that in the inositol 1,4,5 trisphosphate, an extra phosphate group is present at the position number 1 of the inositol. So therefore it is known as the inositol 1,4,5 trisphosphate, and this trisphosphate actually refers to 3-phosphate. So for short, this diacyl glycerol is known as the DAG, which actually remains in the cell membrane, and the inositol 1,4,5 trisphosphate is known as the IP3, which is actually a soluble molecule, and it is released into the cytoplasm of the cell. So the activation of the phospholipase C is actually going for the breakdown of the PIP2 into the diacyl glycerol and the inositol 1,4,5 trisphosphate. Now, when the inositol the IP3, when that is released into the cytoplasm of the cell, what it do? Now, the IP3, it goes and it binds to a special receptor on the endoplasmic reticulum, which is known as the inositol triphosphate receptor. And you can actually call this as the inositol receptor, the inositol triphosphate receptor. Now this inositol triphosphate receptor, this is the inositol triphosphate receptor. It is actually a membrane glycocomplex acting as a calcium channel, which is activated by the inositol triphosphate or the IP3, and this particular receptor is made up of 4 subunits, which actually weighs around 313 kilodalton. So if you look at this particular receptor, it is actually made up of these 4 subunits. Now these red spots that you can see over here, that is actually the IP3. So when the IP3, it goes and it binds to these receptors actually open and it allows the movement of the calcium from the endoplasmic reticulum into the cytoplasm of the cell. Now initially what happens is that the concentration of the calcium that is kept very low in the cytoplasm, because the high calcium concentration in the cytoplasm can create various physiological problems. So usually the calcium concentration in the cytoplasm that is kept low, and inside the cell the endoplasmic reticulum is acting as a store for the calcium. when this calcium is needed, that is then released from the endoplasmic reticulum into the cytoplasm to perform their physiological function. So if you see over here, when a ligand comes and it activates the phospholipase C, the phospholipase C then make this IP3, and the IP3 binds to this Inositol Triphosphate Receptor, opening this channel, this calcium channel, which actually allows the movement of the calcium from the endoplasmic reticulum into the cytoplasm of the cell for their functions. Now, when you talk about the high calcium concentration in the cytoplasm, that is not desired. So what happens is, when the concentration of the calcium that rises in the cytoplasm, there are mechanisms which actually drop down the calcium concentration in the cytoplasm. Now, one of the important mechanisms that is there, that on the surface of the endoplasmic reticulum, there is a particular calcium ATPase, which is known as the SERCA. Now, the SERCA is actually the sarcoendoplasmic reticulum calcium ATPase. Now, what this SERCA do is that it is going to transfer the calcium from the cytoplasm back into the endoplasmic reticulum. And how it do is that the SERCA, it actually allow the movement of the calcium from the cytoplasm into the endoplasmic reticulum, utilizing the ATP as a source of energy. So during the transfer of calcium from the cytoplasm into the endoplasmic reticulum, a molecule of ATP that gets converted into the ADP, an inorganic phosphate, and this degradation of the ATP actually provides the energy which drives the movement of calcium from the cytoplasm into the endoplasmic reticulum. This is one mechanism by which the high calcium concentration of the cytoplasm is dealt with. We call this as the calcium ATPase because it is actually responsible for the transfer of the calcium. And when we talk about the ATPase, so ATPase are actually those channels which utilize the ATP as a source of energy. By degrading the ATP, they are getting the energy, therefore they are known as the calcium ATPase. Now, the movement of the calcium from the cytoplasm into the endoplasmic reticulum through the SERCA is not enough to control the high calcium concentration in the cytoplasm. So there are other mechanisms as well. If you look at the cell membrane of the cells, so at the surface of the cell membrane, there are special calcium channels which are known as the PMCA. And this PMCA is plasma membrane calcium ATPase. So the SERCA and the PMCA, both of them, they are calcium ATPases, but their location that is different. The SERCA is present on the endoplasmic reticulum. The PMCA is actually present on the plasma membrane. So on the cell membrane or on the plasma membrane, there is this PMCA, and again this is an ATPase. This means that it will be driving the movement of the calcium from the cytoplasm into the extracellular matrix, utilizing the energy of the ATP. So in ATP, that will be converted into the ADP and the inorganic phosphate. So as the concentration of calcium is high in the cytoplasm, so the PMCA is actually helping to remove the calcium from the cytoplasm into the extracellular matrix, so that the high concentration of the calcium in the cytoplasm is dealt with. But one of the important difference between the PMCA and the SERCA is that when the PMCA, it moves one calcium from the cytoplasm into the extracellular matrix, on the same time it also brings two protons inside the cytoplasm, so that the charges on both sides that remain balanced. Because if you are removing this positive charge from the cytoplasm into the extracellular matrix, you may change the electrical potential of the cell. Therefore, when the PMCA, it removes one calcium, it is having two positive charges. When it removes one calcium from the cytoplasm into the extracellular matrix, it brings two protons which actually have two charges over here. So you are removing two positive charges, you are getting back two positive charges. So the charges on the inside of the cell, that remains the same, the interior and the exterior of the cell. So this is the second mechanism by which the calcium concentration, the high concentration of the calcium in the cytoplasm that is dealt with. Another strategy is that some of the calcium that do binds with certain proteins in the cytoplasm, and one of the important protein that calcium is going to bind is known as the calmodulin, and we will have a detailed discussion on the calmodulin, the interaction of the calcium with the calmodulin, and then the function of the calcium calmodulin complex in the videos to come. But these are actually the three strategies that are usually utilized to control the high concentration of the calcium in the cytoplasm. One, they are moved from the cytoplasm into the endoplasmic reticulum by the Sarca. They are removed from the cytoplasm into the extracellular matrix by the PMCA, and some of the calcium they go and it binds to the calmodulin, making the calcium calmodulin complex. Now, if this process continues, say for example ligand comes and it activates the Phospholipase C, the IP3 release the calcium from the endoplasmic reticulum, and the PMCA is removing the calcium from the cytoplasm into the extracellular matrix, there will be a time when there will be no calcium in the endoplasmic reticulum and the signaling pathway that will be dead. So how the endoplasmic reticulum that actually deals with this? Because if you are releasing the calcium from the endoplasmic reticulum, you are not getting that much calcium back into the endoplasmic reticulum which has been released. For example, if you are releasing 10 calcium ions from the endoplasmic reticulum, with the help of the Sarca, you may be able to get only 4 inside the endoplasmic reticulum again. What this means is there will be depletion of the calcium ion. So when there is a depletion of the calcium ion, what happens is that there are proteins present inside the endoplasmic reticulum which are known as the stem 1 and stem 2. Now initially when the calcium concentration inside the endoplasmic reticulum is as per desired, these stem 1 and stem 2 that remain in the monomeric form. But when there is a depletion of the calcium in the endoplasmic reticulum, the stem 1 and stem 2, they go and they make these oligomers. They combine with each other, making these oligomers over here. So if you can see here, it is in the monomeric form, but in case of the calcium depletion, they actually combine with each other, making these oligomers. What these oligomers do is that they move from the endoplasmic reticulum into the towards the cell membrane, and there they bind to specialized channels which are known as the SOS or SOCE channels. Now this SOCE is actually the store operated calcium entry. Now this store operated calcium entry, it contains channel proteins like the ORAI1, 2 and 3. So what happens is when there is calcium depletion in the endoplasmic reticulum, the stem 1 and stem 2, they are going to make these oligomers. These oligomers that will move towards these SOCE, and when these oligomers they bind to the SOCE, this particular calcium entry channel, it usually opens, and the calcium that moves from the extracellular matrix into the cytoplasm of the cell, because the concentration of the calcium is now high in the extracellular matrix, it is low into the cytoplasm. So the calcium that will be moved with the help of the SOCE from the extracellular matrix into the cytoplasm of the cell. Now when the calcium it gets into the cytoplasm of the cell, some of the calcium that will enter into the endoplasmic reticulum with the help of this Sarca, and some of the calcium that enters through the SOCE that will be moved again by the help of the PMCE from the cytoplasm into the extracellular matrix. Now this process continues, the entry of the calcium with the SOCE, the entry of the calcium into the endoplasmic reticulum with the help of the Sarca, and the removal of the calcium from the cytoplasm into the extracellular matrix. This process continues until the stores of calcium in the endoplasmic reticulum they are replenished and they are taken to a level which is the desired one. So this is actually something about the calcium homeostasis, how the concentration of the calcium in the cytoplasm and in the extracellular matrix that is actually operated. So when this calcium when it this is the homeostasis which is actually carried out by all of these strategies. Now there is a particular drug which is known as the tepsigargan, and this pepsigargan is actually an inhibitor of the SERCA, and this is actually an experimental drug which is used in the studies where you want the inhibition of the SERCA, this particular channel, and you want to study the effect of the SERCA on the calcium homeostasis. Now, when the calcium and the this homeostasis is maintained, the next step in the pathway, that is the activation of the protein kinase C, and we will continue the discussion on the protein kinase C in the next video. So if you like the video, please share to my channel, hit the like button and share it with your friends.